drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode.
Thank you. Thank you.
Can you start off by just telling everyone a little bit about who you are and what your lab does?
Yeah, so I'm a professor at the University of Trento, Northeast Italy, where I lead a lab which is studying the human microbiome, so all the collection of microbes populating our body. I started to be interested into that more than 15 years ago now, when I joined in the US the Human Microbiome Project. And after that, I've been always more and more fascinated with this topic that we are studying mostly through this approach called metagenomics. And now I lead this lab applying metagenomic approaches mostly on the human microbiome for trying to discover both basic characteristics of the human microbiome, but also aspects that are related with health and disease.
So I know what the Human Genome Project was, but I don't think I've heard of the Human Microbiome Project. What was that?
Yeah, it's actually a bit related, no? Because it was 10 years after the Human Genome Project. At that time, researchers started to appreciate the importance of the human microbiome. So they said, let's sequence whatever is not human inside us, and let's get a map of the human microbiome instead of the human genome. So it started around 10 years after the Human Genome Project. However, it was much more difficult because the researchers realized pretty soon that it was impossible to have a very clear and specific map of the human microbiome, mostly because there is much higher variability in the microbiome than in the genome. So it was difficult to do that. And so the goal of the project changed a little bit toward trying to investigate what is normal to have or not to have in our human microbiome overall.
Yeah, and I suppose that's a huge problem, because it's not simply that there's variability from person to person and that there's many species of microbes, but you've got many different genomes that vary at different locations, even within a single individual.
Yeah, there are really tons of challenges there. There is a lot of variability. Yes, there is a variability between individuals, but also between populations. And there are a lot also of microbes that we never heard about. Microbiologists never cultivated, especially at that time, most of the members of the human microbiome. And so it was difficult to make sense of it. However, what helped a lot was metagenomics, because until the use of sequencing-based approaches, the only way to study the gut microbiome and the human microbiome in general was to try to cultivate the single members. But that is incredibly difficult, and sometimes it's really impossible, because you cannot know, maybe one microbe is interacting with another microbe, so and this interaction is vital for both of them. So you cannot
separate
cultivate
them,
only one of them. And so metagenomics, at least from the biotechnological viewpoint, started to solve this issue. So the idea was, let's sequence the genetic content of a sample full of microbes, and by analyzing the genetic content, we can have an idea about which microbes are there.
Mm-hmm.
but this is difficult, no? Because, the idea, like with the Human Genome Project, the technology is mostly the same, sequencing the DNA. But sequencing the DNA in the case of the Human Genome Project was only the first step. Then we had to make sense, to reconstruct the full genome, starting from readings of short pieces of the genome. So this is called genome assembly. And is the idea is like a big puzzle, not trying to put together little pieces to reconstruct the full genome. But with the microbiome, is much, much more difficult because it's like trying to solve many, many puzzles all together, starting from the pieces of the puzzle all scrambled together, and we need to reconstruct many, many puzzles, and each puzzle is the genome of some of these bacteria, and some of these bacteria are completely unknown. So trying to reconstruct a puzzle without having the picture of what is the final goal.
I see. So the genome is like a puzzle, like, literally a puzzle that you could build with your family on the tabletop. And of course, the pieces are all mixed up, but it comes with a box. So you know what the picture looks like. And I guess with the Human Genome Project, we had a sense for what the human genome looked like. We knew the number of chromosomes. We sort of have the picture on the box. And so that helped in assembling that one genome. But you're talking about metagenomics, which I suppose refers to the fact that, inside of us, there's many different species of microbes in the microbiome. And so you're saying it's akin to having, a bunch of different puzzles all mixed together. And then on top of that, you don't even have a box for many of them because they are brand new species and we have no idea what they're supposed to look like.
Correct. In the gut microbiome, there are usually many hundreds of these different bacteria. And not only bacteria, there are also viruses, fungi, and so on and so forth. So these are the number of different puzzles that we want to reconstruct, and some of the genomes are quite similar. No, it's like the blue sky of many different puzzles. The blue is kind of the same, so it's very difficult even to assign the single fragment of DNA or the single piece of the puzzle to the specific final picture you are trying to reconstruct. So this is to say that basically what kept us busy, our lab and many other labs, was not trying to improve the biotechnological approach of the sequencing, but more analyzing, developing ways to analyze this data. And so we move from being able to know which phyla, so which large taxonomic categories were there, to arrive to specific species or even specific strains or even specific variants. So, distinguishing the nucleotides that are making different, that are differentiating the Escherichia coli in my gut and in your gut, for example.
So it sounds like when you're doing this metagenomic work, the data acquisition side is sort of solved. You have the technology, you can do the sequencing. The problem and the challenge and potentially the interesting thing for someone like you as a computational biologist is really on the genome assembly and data analysis side.
That is the point. That's the reason why I'm a computer scientist and I can apply computer science approaches into this field. It's not only about assembly, because assembly is probably the easiest approach we can think about, but there are many other tasks that we can achieve that are different from assembly. But yes, that is exactly the point. On the technology, the only thing that changed a lot is the cost for sequencing. 10 years ago, it was about 1,000 euros to sequence a sample, a metagenomic sample. Now is like 10 times less expensive. And this means that we can have much, much larger data sets and codes sequenced, which is at the basis of the big findings, because again, the microbiome is hugely variable, so to make sense of it, you really need to have large sample size.
Yeah, and it's not only variable in the ways that we've mentioned so far, but it's dynamic in a way that the genome isn't. So even though the gene, like, of course, our gene expression can change, genes can turn on and turn off within an individual, but despite all of the expression differences, there's still one genome in all of my cells that's the same genome. But that's probably not the case with the microbiome. The species are probably coming and going, shrinking and expanding to different degrees depending on lifestyle and a bunch of other factors we can talk about. So how do you start to how do you start to like standardize things in the lab so that that you're kind of looking at the same thing? Like, do you have to make sure and be very careful about taking samples from the same person in the same state? How do you sort of handle the dynamic nature of the microbiome?
Yeah, that's a fantastic question. So, yeah, in the lab, we need to be careful for many reasons. So, for example, contamination. Microbes are everywhere, in the air, in the kits, on the bench that you have in the lab. Even if you are trying to stay as sterile as possible. So microbes are everywhere, and if you don't pay attention, they will end up in your experiment. On the variability and longitudinal variability of the microbiome, we did a lot of studies, and I think we appreciate right now that the single components in your gut, in your oral cavity, are pretty stable in time in terms of which microbes are there. However, they change a lot in terms of abundances of single microbes, and the question that we started to be extremely interested in is how do we acquire such microbes? No, so if you sequence my gut microbiome, if you sequence your gut microbiome, I'm sure we will have around 20 or 30% of species in common. It's just a little bit if you think about that, no? Because it's like less than half of the species in my gut are also in your gut. But even more astonishing is if you look at the genome of these microbes that we have in common. So I have a Escherichia coli in my gut, you have a Escherichia coli in your gut. But if we look at the genome, they are only 95% or so similar on the shared genome. So many genes my E. coli has it your doesn't have, and vice versa. At the point that we really think that each human microbiome is unique if we look at the genetic of the human microbiome.
So we'll have some species in common, but the majority of them won't be in common. And even for the species that we both have, we both have E. coli in our gut. Basically, what you're saying is they can be different strains of E. coli. So even though they're the same species, they're not really identical.
For E. coli, for example, there are strains that are probiotic, strains that are pathogenic, strains that are commensal, strains that are carcinogenic even, or environmental. And so, knowing which strains we have is important, and that's what actually we are trying to study. And the next step is to understand how do we acquire these strains from. So the baby in utero is sterile, and so instead, a baby of few days of life is already full of microbes. And so we started to focus there to understand what is the process of colonization for our gut and human microbiome in general, and that is what we started several years ago by looking at. We went into the hospital and we sequenced the microbiome in many different locations of mothers before they were giving birth, and then the baby just after birth. And we really saw that basically the strains that were present in the mother were those present also in the baby. So with a vertical transmission from mothers to infants that were occurring during delivery. And so, this is to answer your question on what's the variability and longitudinal variability. We start setting a first, kernel of microbes at birth from the mother usually, and then we continuously change it in time.
And so, when the baby is still in the womb prior to birth, you're saying they don't have a microbiome; it's sterile. Do you mean inside the baby, like the gut microbiome, or even is the entire womb devoid of microbes?
It's entirely womb. There are exceptions. Some cases there are some potential pathogens, but in healthy pregnancies, they are sterile. Now, sterile or almost sterile is a small difference.
Very, very low microbe.
Very low. So there is not a working microbiome in the baby before birth.
And so is it literally like a physical process? Like the baby is born, it's going through the birth canal, and it's touching the mother's tissue as it's rubbing against it as it's being born, and that's literally what seeds the microbiome.
That's exactly how it happens. And in fact, we see that when there is the C-section instead of natural birth, this seeding of the microbiome from the mother to the baby is not occurring. It's not a very, very big deal because it can still happen in the days after delivery by direct contact. But yes, this is a confirmation that the seeding starts already at birth with the vaginal microbiome, but also with the gut microbiome, which is in any case contaminating many body sites. And we do see this happening. And again, the confirmation is that in the baby, we see the microbes coming, the same exact genetics of the microbes in the corresponding mother, which are different from the strains we see in any other mother. So it has to be at that level of transmission.
Mm-hmm. And so after that microbiome is seeded and it sort of takes root and establishes itself and becomes stable after birth, I know that the microbiome is plastic. It can change, but how difficult is it under normal natural circumstances for that microbiome to change? So I guess one way of asking that would be, so if a baby's born, it gets its microbiome. If you look 6, 12, 18 months later, 5 years later, whatever, is it more or less going to be the same microbes with the same composition as right after it was born? Or is it going to be completely different? Or is it somewhere right in the middle?
Well, it depends at the age, So infants at birth, they change very rapidly the microbiome. Then they go through changes like breastfeeding or starting to eat food and so on and so forth. So, in the first 1.5 years, the microbiome is changing a lot, very rapidly. Then it changes less rapidly, and in the adult is relatively stable at the point that usually, if I look at my microbiome now and my microbiome in a year from now, if I didn't do any big changes in diet or didn't have any medical condition or change content or whatever, we expect to see 80% of the strains to be the same. But there are multiple things you can do to change your microbiome because you want to do that or because it happens. So diet is one big way to change your microbiome. Lifestyle, drugs, of course. For many, think about antibiotics, for example. And then you can arrive to medical ways to change the microbiome, including, for example, fecal microbiome transplantation, which seems to be quite a dirty thing, which is actually what it says. So taking the gut microbiome from an healthy individual, giving it to a diseased individual, and this is a way to reprogramming the gut microbiome quite efficiently, actually.
Yeah. So the microbiome is plastic. It changes quite a bit early in life, naturally. Just, there's a lot of change happening early in life, so it's pretty intuitive. But it stabilizes by the time you're an adult, such that, unless you make major changes to your diet, to your lifestyle, you start taking antibiotics or other things, it will be relatively stable from day to day, from week to week. In a stable, healthy person with a consistent diet and lifestyle, an adult, I'll let you define what exactly this means, but the more important or more consequential microbes, if you were to look at the 1 or 2 or 3 biggest strains in terms of how much they're influencing the physiology of the person in terms of their population density or whatever's relevant here, is there any sort of general principle in terms of like what they're doing? For example, are they primarily preventing pathogens from getting into the body by sort of taking up that space in the gut, or are they primarily digesting critical food components for us? Is there any pattern there, or could it be sort of any of the above, depending on the specific species?
All of these all together. So an healthy, diverse, and stable microbiome is doing a lot of functions, preventing infection, as you mentioned, but also helping us digesting food, producing some compounds, short-chain fatty acids, or some vitamins that they will not be available to us in any other way. But also, priming and setting the immune system is an extremely important function that has implication even on tumors, for example, because an immune system in a positive and healthy configuration can prevent diseases from autoimmune diseases, but also to cancer and much more. and then there is the gut-brain axis, for example. So the interaction between our gut and our head by this gut-brain axis. So, the function of the human microbiome are very, very big. Metabolism much, much more, and an healthy microbiome does all these functions all together. Also, though some that I am sure we still have to discover completely.
Mm-hmm. So given how prevalent antibiotics have been for many years now in our society, it's pretty well known that this is obviously affecting the microbiome. It's not just affecting the pathogens that we intend to use the antibiotics against. But if you take broad spectrum antibiotics, you're basically clearing out your microbiome or greatly reducing it or affecting it in some way. When that happens, given that it's happened to many, almost everyone has taken an antibiotic at some point in their life.
Yeah.
But when that happens, so let's say I take an antibiotic tomorrow, a high dose of a general antibiotic, and then I get better. I'm no longer sick, and then I recover. How likely is it that my gut microbiome is going to recover and be the same basic population it was before, or is it very likely to be completely different?
Yeah. So antibiotics are clearly a huge problem, especially for inducing pathogen resistance. So some pathogens become resistant to antibiotics because of the use of antibiotics themselves. But instead, in terms of the microbiome, yes, you kind of reset the microbiome, and then the microbiome, after you end taking antibiotics, starts to rebuild. I wouldn't say from scratch, but from the seedings from the seeds that remain there.
Mm-hmm.
And I would say that most of the time, which we can give percentages here, probably between 70 and 90% of the times, you go back to an equilibrium of your gut microbiome, which is similar to what you had before.
Mm-hmm.
But the problem is that 10, 20, 30% of the cases in which you reach another equilibrium can still be a good equilibrium, maybe in some rare cases even better than the original ones.
Mm-hmm.
But in other cases it's not. And so this may induce allergies or other problems that are due to that. However, I don't think actually antibiotics are the drugs that with the worse impact on the gut microbiome, because
Really?
antibiotics, you take them for a couple of weeks maybe, but then you stop them. There are other drugs that are you assume them for years or even forever. For example, proton pump inhibitors, they are changing the pH of your stomach, and they are allowing many different microbes arriving to your gut. And it is something that is not like, again, 2 weeks like antibiotics, but is for many, many months. And this is impacting much more the microbiome.
Yeah.
So we can find from the microbiome, we can predict whether you are under proton pump inhibitors at very, very high accuracies. So there is a signature of it.
And I would imagine something like a proton pump inhibitor, it's changing pH. That's a very broad spectrum stimulus. It's going to basically affect any microbe species that would be there no matter what.
Correct. And also changing the acquisition of microbes, no? Because the way we change our microbiome, coming back to the previous topic, how is our microbiome changing? So if you think our microbiome, the members of our gut microbiomes are not living in the environment. It is not that, on this table, or if I go into the woods, the microbes that are there are also the microbes in my gut. 99% of the microbes in our gut can only be in the gut of other individuals, or maybe other primates, but we are not in touch with primates, no? So basically, a study that we did recently really showed that is the person-to-person interaction leading to the exchange of microbes and the changing of our microbiome. So we discussed before, mother transmitting microbes to the infant, but even as adults, we interact, and as we very well know, that we exchange pathogens, from viruses, SARS-CoV-2, whatever. We exchange also the other microbes.
Yeah.
And we know, in a household, when a pathogen arrives, SARS-CoV-2, for example, 99% of the cases it gets to the whole family.
From one person to the next,
it's the same with our gut microbes. And that is the way we are seeing. So, for example, if you take two individuals like you and me that we never interacted directly, we probably don't have even one strain in common. But if we start to live together into a house for a couple of years, statistically, we'll arrive to share 20% of our gut microbiome, even just because we share the same physical space.
I see. So a couple of things pop up for me there. So it sort of makes intuitive sense that if you share a common space, a common environment with other people, your microbiomes will converge to some extent. So there's a sort of a two-pronged question there. To some extent, that could just be shared environment. We're both eating the oranges on the table, the same foods in the fridge. We're naturally going to converge on diet to some extent. So naturally, that will facilitate a convergence to our microbiome to some extent. There's also the question that you brought up of direct transmission. Can I catch my gut microbiome from you literally? And I want to ask you about that. So again, if we think about other pathogens, respiratory viruses, things like that, very simple, very intuitive. If someone has a respiratory illness, they sneeze or they cough, I'm going to inhale those viral particles or whatever, and it goes from their lungs to my lungs. It's a little bit more difficult for me to think about how this happens with something like the gut microbiome because, as you said, many of these species are adapted to live in my intestine. They probably can't, I would imagine, they can't live on my microphone. They can't live on my hand. They can't even live in my mouth if they're adapted to, say, my colon or my small intestine. How does one transmit their gut microbiome if that's the case?
Yeah, so that is a fantastic question. So we know very little about that because we just discovered recently that there is this huge transmission of the microbiome in general. But first of all, the oral microbiome is more transmissible than the gut microbiome. So I said that if we stay together for 2-3 years, we share 15-20% of our gut microbiome, but we are probably going to share 40-50% of our oral microbiome.
I see.
Well, this actually measured when is partners that are also kissing, but even in any case, sharing the same space will lead to higher sharing of the oral compared to the gut microbiome. But if you think still of the pathogens, even bad gut bugs are transmitted almost in the same way, with the same efficiency that the oral pathogens or the respiratory pathogens. So, if you go back even many years, microbiologists developed this theory that all microbes are everywhere, and then the environment selects. This can be a specific application of this theory. So, because we put our microbes everywhere because we just need maybe few spores or few cells of gut microbes that are in the environment, and this will be enough to have the microbe, if it arrives into your gut, to find the perfect environment to actually grow. So when we study transmission, we are actually unable at the moment to disentangle what is the effect of my microbes arriving to your gut and the probability that transmission will actually result in a long-standing colonization. And without being able to disentangle these two effects, it's going to be very difficult to understand what are the main sources and modalities of transmission.
Interesting. Yeah, it makes sense. I mean, obviously, if the baby picks up the microbiome from the mother through direct contact, I mean, people who live together have lots of direct contact together, especially, if you imagine your spouse or your partner, you have direct mouth-to-mouth contact, and that's going to influence the composition of the microbiome.
But you have to, the microbes transmitting between adults, they need to break what is called colonization resistance, because each of us, you have your microbiome, which is well established. It has an equilibrium. It's not so easy for someone to arrive and to kick one strain out and colonize there. Yeah. The baby is favored by the fact that it's not colonized by anything, so it's much more likely that whatever arrives there will actually colonize. And this is the same with antibiotic resistance. So we saw that babies at the daycare, after they take antibiotics, they are much more prone to acquire microbes from their peers, because basically they are resetting the microbiome, they are lowering the colonization resistance. So the other incoming strains are much more likely to colonize.
Wow. And to the extent that the microbiome that's transmitted to you can cause you to develop or lose a given phenotype or trait or health outcome, it seems plausible that in human society today, the dual use of drugs like antibiotics or like other drugs, like you mentioned, proton pump inhibitors, plus the interactions we have with different individuals, you could essentially catch things from other people if you become prone to acquiring the microbiome, if you just went through an antibiotic treatment or some other drug treatment, for example. And I guess maybe one way we can sort of start to talk about this is, I know that there's work out there, for example, showing that in rodents at least, you can basically transmit obesity from one mouse to another. If you give the obese mouse's microbiome to the lean mouse, and the lean mouse becomes obese. And those types of experiments have been done for obesity and other things, as far as I'm aware. To the extent that's true in humans, that means, in theory, right, you could have a nice, healthy, lean baby, say, or child. They get sick, you give them antibiotics, you sort of clear out or diminish their microbiome, and then they go to daycare. And if they're around someone, another child who's obese, they could catch their obesity. Is that possible? To what extent do we think something like that is actually happening?
Yeah, we need to be a bit careful here. But for sure, what we know is that our social interaction and our lifestyle is impacting our microbiome by really, the people we interact with, because we are catching the microbes from there. And yes, there are several diseases that are linked with the microbiome. However, if it is not a single pathogen, there are very, very few diseases that are only due to the microbiome. Okay. So it is a complex system. But yes, for some conditions that are causally linked, at least in part, with the microbiome, then yes, microbiome transmission makes these diseases that are usually called non-communicable diseases at least partially communicable. And that is actually what we are studying right now, because as I was telling you, we started discovering this microbiome transmission, and now also other scientists added exactly your same idea. Maybe part of these non-transmissible diseases are actually transmissible. We are studying this. We are studying it from cancer to obesity to metabolic diseases.
Yeah, because normally, this is the definition of a non-communicable disease. You don't normally think about catching cancer from someone or catching metabolic syndrome from someone. But what we're talking about here is the possibility that there's a context-dependent possibility that otherwise non-communicable diseases can become communicable. And the microbiome...
I would say that maybe the risk for the disease becomes a bit communicable. You mentioned cancer. There are some cancers that are caused by microbes. And so in that cases, it is known. But these microbes are usually not pathogens, are microbes that are enhancing the risk for developing certain kind of cancers. So it could be that if you live with a person that has these bacteria that are slightly more prone to develop cancer, then maybe you can get them.
And how exactly does that work? Are these microbes producing a metabolite that is mutagenic or something like this?
Well, in the case of cancer, there are many different ways. It can be by inducing inflammation, and inflammation in turn may be a risk factor for cancer. There are some microbes that are known to indeed basically damage the human genome, and that can cause cancer. And there are other bacteria that are thought to be protective against cancer, and so not having them may be an indirect way to enhance the risk for cancer. The microbiome in oncology is a relatively novel topic, and relatively means probably 5 to 10 years. Again, if we exclude the pathogen part, and this is very, very quickly developing. Also, because we have to think about the microbiome as also something to exploit for treatment. I'm referring, for example, to the cancer treatment. The immunotherapy is one of the latest fantastic discoveries in the field of oncology. Immunotherapy is saving life of people right now. So people with melanoma, for example, advanced melanoma, that are not responding to any other chemotherapy or other therapies, some of them, a good fraction of them, are responding to immunotherapy, completely cleaning out the cancer. But in some cases, this is not working. And we saw that the microbiome, in some ways, is linked with responding or not to immunotherapy. And this is extremely important. And to prove that, some very recent trials did fecal microbiota transplantation from an healthy donor to someone who was not responding to immunotherapy, and after reprogramming the microbiome via FMT, fecal microbiota transplantation, some of these individuals started to respond to immunotherapy.
So the immunotherapy is not working, and then you change their microbiome, and then the immunotherapy is now effective, and it wasn't before.
Correct. So this is linked with what you were saying before. No? So FMT means basically inducing a transmission, is artificial transmission. Is not by being taught in contact with someone, is really, artificial transmission. And this induced some changes in the microbiome and in turn in the immune system, which made immunotherapy starting to work. And this is proven and is more and more an important topic in oncology.
Do we know exactly how that works? Is the new microbiome stimulating the immune system to operate in some new fashion? Is it producing a molecule that wasn't there before?
Yeah, the answer is that we don't know, unfortunately. And that's the reason why we do FMT, which is transmitting everything. Because if we knew which are the features and the important thing, we would just transplant that, or basically having some new probiotic kind of intervention targeting directly that. So clearly, of course, there is a lot of research here because we want to understand. But my feeling is that this is not going to be very successful because our human microbiome, as again and again, is unique to each of us. So even the intervention that you will need to do it on each of us will be unique. So
going back to the subject of diseases that are clearly linked, at least in part, to the microbiome, what would be one of the best examples where the link is clearest between a disease state and the microbiome?
Well, if we remain in oncology, for example, some colorectal cancers are known to be linked not only with some specific bacteria, but also with some other bacteria that are colonizing the tumor. So this is one example. Of course, in all the metabolic diseases, is not a primary cause, but is one of the causes and one of the things that being unbalanced is leading to the disease. And of course, also all the autoimmune diseases have a seed in the microbiome.
And so to what extent, I'm not super familiar with the literature on this, but whether we're talking about something like metabolic syndrome or something like an autoimmune disease, to the extent that any one condition is linked to the microbiome in some substantial way, in theory, I would imagine. So as you said before, right? You're as an adult, your microbiome reaches some kind of like equilibrium state, and it kind of stabilizes depending on what your lifestyle is. So it's easy to imagine that some people with certain ailments might sort of be stuck with a microbiome that's suboptimal, but it's stable in their bodies. And so that would imply that a potential strategy could be that if you sort of just take, say, broad spectrum antibiotics or something, wipe out the microbiome, and then reset it intentionally with, probiotics or a particular diet or a direct transplantation from another individual, this could be an effective way to reset the microbiome. And I mean, is anyone doing anything like that? Have we seen examples of something like that where you sort of intentionally reset someone's microbiome?
Yes, I guess we know it that after antibiotics, having an healthy diet is particularly important to recover our health, and which is probably also due to the microbiome.
Mhm.
Probiotics is another topic. Yes, probiotics. Many of the probiotics are not directly colonized in our gut, so it can be a bit more complex there.
Mhm.
But yes, we can arrive to the point of thinking about doing auto-fecal microbiota transplantation to recover our microbiome before after having a disease. Although for this we will need to be very forward-looking, because we will need to store our microbiome for future transplantation, auto-transplantation, before getting the disease, because otherwise, we don't have the healthy microbiome anymore.
Yeah. Another thing I want to talk about is um, ancient microbiomes, because I believe you've done some work here. So, by analogy, we all know that genomics is a field, and you can get your genome sequenced today as a living person. But also, ancient DNA and ancient genomics has been a very vibrant field for the past, X number of years. People have sequenced Neanderthal genomes and so forth. It sounds like you've been involved in work where you can actually recover aspects of the microbiome from deceased individuals, going back quite a ways. To what extent is this possible, and what did you find there?
Possible. So our interest in the ancient microbiome started when we studied the difference between our microbiome and our being the westernized, urbanized, American, European urban microbiome, compared to what we call non-westernized microbiome. So population living in a very rural area, hunter-gatherers as an extreme, but in any case, not having access to drugs, to high-fat diet, and things like that. And we saw completely different microbiomes. But at that point, we didn't really know whether it was the lifestyle that was inducing the difference in the microbiome, or was some other geography associated characteristics. So to answer to this question, we basically try to go back in time and look at mummies, the microbiome of mummies, in the same exact area we were living. And we were lucky here because on the Alps, close to the border between Italy and Austria, 25 years ago or so, it was found a very ancient mummy. It's called the Iceman, or Otzi. Is 5.3 thousand years old. Is extremely well conserved. So it's possible actually to go into the gut of the individual, and we know what was the last meal, but we also have the microbiome content of it. And so when we sequence it, and this was difficult, but was supported by the scientists, the anthropologists, and the microbiologists working on this specific kind of samples, we actually found a lot of bacteria that were almost identical to the one of non-westernized population, and completely different from our own modern urbanized populations.
So the genetic material of the microbiome was intact in this mummy.
I wouldn't say that it was intact. The DNA was slightly more fragmented, but it was possible to identify the strains with high precision. And we were also favored by the fact that, as we were discussing, the gut microbiome is only present in the gut, so we could distinguish very well the gut microbes with respect to contaminated soil microbes, for example, because, of course, this guy was found in the environment, so on and so forth. But yes, even though ancient DNA have some specific patterns of damage, we were able to reconstruct a large fraction of their microbiome.
Wow. And I'm sorry, what did you say in terms of how similar, how different it was from present-day microbiomes?
So basically, the microbiome in this ancient mummy was much closer to current modern non-westernized population than our own westernized population.
I see. It looked more like someone living in a rural area today than an urban area.
Exactly. It was very similar to the hunter-gatherer population in Africa, for example.
Huh. Oh, interesting.
Yeah, compared to the population in Trento, which was only 60 kilometers or so from where the Iceman was found.
Mm-hmm. And when we compare present day microbiomes on average from like the Western urban microbiome to a more hunter-gatherer style microbiome. Are there any general patterns there in terms of the differences, like where, in terms of how the difference is allowing for certain nutrients to be absorbed that are in the one diet but not in the other, or are there any sort of patterns as to what the functional difference between the microbiomes actually is between a hunter-gatherer and an urban person?
The biggest difference we see is that hunter-gatherers have a lot of microbes and genes in those microbes able to degrade very complex vegetable fibers that probably we don't get usually in our diet anymore. But this is only the biggest difference. There are many other differences. For example, we see less genes having to do with pollutants or xenobiotics in general in the westernized population. But what is more relevant, because you may think, sure, we don't get very complex fibers in our diet right now, so who cares not having those bacteria? But we have to think about the coevolution between humans and our microbiomes. So this, it was a process going on for millions of years through the primate evolution and everything. And so the microbes that are there don't have only one single function. So the fact that we are losing the bacteria that were there evolutionary because we are not eating complex fibers anymore has a lot of impact, because those microbes are there and learn how to stimulate in the right way the immune system, how to do many other functions, and losing them may be a big problem. So we are studying that. We need to be careful, but this may be linked, for example, with the increase of autoimmune diseases or other allergies or intolerances that are in our population right now. This may be due to the disruption of the host microbe coevolution due to the change in lifestyle.
Mm-hmm. Mm-hmm. What about other medications that people take chronically? And there's many things we could potentially talk about, but it makes sense that obviously antibiotics affect the microbiome. It makes sense what you said before about proton pump inhibitors, because they're just going to change the pH, which is going to be, it's going to favor and disfavor different species in the gut and elsewhere. But what about other types of drugs? Because a lot of people are on so many pharmaceuticals now, and I could imagine, for example, just to give one example, SSRIs. They're very common. They are affecting serotonin. As many people, I think, know at this point, there's a lot of serotonin in the gut. And I would imagine even those types of medications can have an effect on the microbiome. Is that likely the case? Are there any examples where we know that's definitely true?
There are some specific examples, and this is exactly the case. But the point is that this is not as well studied as they should be. But there is more awareness of that in both directions, also because the effectiveness of certain drugs are impacted by the gut microbiome. For example, there is a quite well-known molecule, digoxin, which is a cardiac drug, that there are some strains of a species called Egertella lenta that are able to deactivate completely, so digest basically this molecule, which is not anymore effective in individuals with that. So the interaction between drugs and our microbiome should be studied much more in both directions. Sometimes certain drugs would not be as effective as they should be because of the microbiome. In other cases, the drugs are changing the microbiome in such a way that then the microbiome is in turn causing some diseases or some side effects. And this is still largely uncharacterized. And there are several labs actually now starting to study it extensively. So at the level really of testing each single drug with each single microbe and then building a huge map basically of the potential interactions.
Mm-hmm. Yeah, so it makes sense that there would be, so it sounds like we have examples where, a drug that would be effective is not effective in certain individuals because they've got some microbe inside of them that metabolizes the drug before it can actually be beneficial.
Yeah, the one I said is one of those examples. it can also be vice versa. For example, there is this condition called auto-brewery syndrome. These are caused by some bacteria in their gut that are actually fermenting sugar and producing alcohol instead of producing lactic acid, for example. They are actually doing what is currently done for producing beer or wine. And these individuals that are pretty rare, but they are documented, are basically producing alcohol in their gut, and so also releasing alcohol molecules in the blood. And at the point of, some of these individuals were stopped by police for a normal contact control, and they were actually positive for alcohol, but they didn't drink anything. It was auto-brewery syndrome. So,
Wow.
alcohol here is a molecule, is not a drug, of course, but is one of the examples through which the microbiome can actually mess up or help us with all these molecules. And drugs at the end are special kind of molecules.
Interesting. Yeah, I would imagine the opposite thing is also possible, where something, a drug, basically like a prodrug, becomes activated and turns into something that's actually an effective treatment only in the presence of certain microbes. Has that been observed?
Yeah, that's a good point. I don't have any examples on top of my head, actually. This is usually more on the good effects now. So again, some vitamins, some molecules, short chain fatty acids that are produced only by the microbiome. But that is another way
What
to which the microbiome can mess up. Yes.
so there's a lot of products out there now for consumers where you can send in a sample of some kind and you can have your microbiome tested and you can see what you have. And then I think a lot of these things will give you things like dietary recommendations or other lifestyle recommendations. Are we at the point yet where you think, like, for the average person, you can find value in getting your microbiome assessed and seeing what's there? Can you take action from that, or are we at the point where it might be interesting to know that, but we can't really do anything with it at this point?
Yeah, this is something important to discuss. I think I think tests can be effective to the extent to which they are asking a specific question. I think it's going to be difficult to have one test and with one test at only one time point without a lot of medical information around you and be able to tell you a lot of things. However, if you have a specific question about diet, for example, or about I don't know, the risk factor for a certain disease, then you can have a system, even artificial intelligence, helping you to answer to that question. And so I will be careful with tests that are not specific for answering to a question, but more general health questions, because yes, they can be interesting in understanding which microbes you have there, but with a one single microbiome experiment without collecting a lot of information about you, there is not so much that one can tell you to improve your whatever you want to improve, or telling you what you need to do or not or not.
One of the studies that was catching people's attention a little bit that you were involved with had to do with the effects of coffee consumption on the microbiome. What did you guys do there, and what did you find?
Yeah, so this is a part of a large collaboration that we have with an initiative that is trying to recommend dietary information based on a lot of readouts about yourself, including the microbiome. So this is one of the examples in which I think it is possible to say something, because we have a specific question, we are collecting a lot of additional information regarding this question, and then we can answer. And in doing that, we build a very, very large cohort database of individuals, right now reaching almost 200,000 individuals in the ZOE predict cohorts, for which we have a very detailed diet data. We have the microbiome sequence for all of them, very deeply sequenced. And also we have a lot of cardiometabolic information about these individuals. So uh, we are using these three set of information to try to link what is the effect of diet, not only directly on health, but also mediated through the microbiome. So what we call the diet microbiome health axis. And in doing that, the first question we asked was, what is the single food with the biggest effect on the gut microbiome? And that turned out to be coffee.
Really?
And I think, yes, it's coffee. And I think there are several reasons for it. The first one is that if I ask you whether you drink coffee or not, you can answer me, black or white, yes or no.
Yeah.
And if the answer is yes, usually then probably you will drink coffee 2 or 3 times per day, every time the same coffee, and the coffee is not
like a binary thing. Yeah, most people drink coffee every day, multiple times a day, or not at all.
Exactly. Instead, if I ask you what's the frequency of you eating broccoli?
Yeah. Sometimes a little, sometimes more, sometimes not. Yeah.
Exactly.
Yeah.
So first of all, it cannot be a huge impact because it's sometimes, and then even quantifying that sometimes is difficult. No.
I see.
But yes, so that is one of the reasons. But so we can tell you basically whether you are a coffee drinker or not based on the microbiome with precision around 95%. Because there is a specific imprinting on drinking coffee on your microbiome. And the biggest effect is on one specific microbe, which is called Losonibacter asaccharicus. Is a complex name. Anyway, is a bacterium that if you're a coffee drinker, on average, you have from 6 to 8 times more abundant in the gut than in non-coffee drinkers. And of course, at this point, one can ask, are you sure that is a direct link? Well, in this case, yes, because we cultivated this bacterium and we saw that it was growing well only when we added coffee into the cultivation media. Wow. So we have also the confirmation there.
Well, do you know either? I mean, what is it specifically about the coffee that's good for this creature? And two, is this species consequential at all? Does it help us, digest something or absorb something easily?
Yeah. These are exactly the two questions that are natural here, and then we are trying to answer. For the first question, we are basically trying to decompose all the components of coffee. There are a lot of, from caffeine to many other metabolites to many other actually fibers. And we want to try one by one to see which of them is stimulating the most. So the other question usually we get is whether this works also for decaf coffee, and the answer seems to be yes. So it's probably not caffeine that is impacting the most this microbe. But this is ongoing studies because we just discovered it, and we have the model set up just now to try to answer to that. For the second question, yes. So we didn't find a lot of direct correlation between the amount of this bacterium and cardiometabolic markers, which was a little bit surprising because all nutritionists are kind of in agreement that coffee is good for health, except if you have any, cardiovascular problem, maybe, but otherwise is pretty established that is good. And so the hypothesis that was maybe this bacterium mediating it, and it's something we still don't know. We don't have strong association right now, but maybe that these associations are just hiding. And yes, with this bacterium in vitro and in models, animal models, for example, we are trying to also understand this. What is the effect of this coffee-stimulated bacterium on the gut ecosystem and also on the gut mucosa?
What so when I think about the gut microbiome, or when I've read about it, typically I see a focus on bacteria. What about fungi? Are there many fungal species that tend to live in the human gut? And is it similar to bacteria where they could be pathogenic, they could be beneficial, they could be neutral, or do they tend to be, say, pathogenic?
Yes, the gut microbiome is composed by 4 main domains. Bacteria, archaeobacteria, like metagenomics, for example, fungi, so what we refer to as microkaryotes, if you like, but also viruses of humans and viruses of bacteria that are called bacteriophages. Fungi, yes, they are very interesting. So usually are not a large portion of the gut microbiome. Probably we never saw them at more than 1% abundance or so compared to bacteria,
I see.
but they are very important. Yeast is frequently present, and many and other microcarriers. Blastocystis, for example, is the microcarrier that we found to be most abundant and prevalent in the human gut. Blastocystis it was considered a parasite, so with this bad meaning. Although we found these microbe much more associated with healthy individuals than non-healthy individuals. So also there was something different from what we were expecting. So yes, also the fungal and microkaryotic fraction of the microbiome is relevant, although usually is not the largest fraction of our gut microbiome. Instead, actually, fungi are much more important in another important microbiome, which is the food microbiome. We studied it quite a lot recently. We sequenced a lot of foods, so things that we eat that are full of microbes, full of bacteria, full of fungi. And in these food microbiomes, actually, the fungal part is much more abundant than in our gut. But these are living fungal organisms arrive in our gut, so there they for sure have an impact.
Interesting. When we think about, if we go back to the beginning, so when the microbiome is first seeded, you mentioned that in a normal birth, the microbiome is seeded by the mother, the physical contact with the birth canal as the baby's born. But if you get something like a C-section, it's a very different story. And so the baby might not be colonized to the same extent or by the same microbes as with a natural birth through the birth canal. Are there any links there between how a baby is born, what the microbiome is seeded with, and subsequent health? For example, are babies born through C-section more likely to develop certain ailments later in life?
Yeah, these are information that are known from literature independent from the microbiome. No, it is known that C-section or not being breastfed, that there is higher chances for some diseases, and that is very well known. Yes. However, we saw that we need to be careful here. So we were talking about antibiotics, we are talking about C-section. These are medical procedures that are saving lives, so is important. But in the seeding, there were several attempts at somehow recreating the seedings in C-section born babies, for example. This was done, for example, by swabbing the birth canal and then swabbing the baby. Also with fecal microbiota transplantation from the mother to the baby. There was a recent study. We also collaborated with Will and Vos that tried this. So put a very, very small amount of the fecal material of the mother in the milk. And this was shown to be extremely effective in repopulating the gut microbiome of C-section babies that were actually completely overlapping with the gut microbiome of babies born by normal natural birth. So these are still very, very delicate and advanced medical procedures, are not clinical practice, but there are attempts at recreating, repopulating, reseeding the microbiome in the right way when that was not possible for several reasons.
Yeah. What? So just to ask you some very general questions. So you were a computer scientist first, and then you got into biology. Is that correct?
Correct. Yes, that was my path.
And I mean, I would imagine just the sort of the biological complexity here, together with improvements in technology over the years that allow us to obtain larger and more diverse data sets than we could before, probably makes this and other areas like this very fruitful territory for people with computational backgrounds to come into biology. So can you talk a little bit about how you made that transition and how it was triggered?
Yes. So when was studying artificial intelligence when it was not that cool to study artificial intelligence. But I was studying it more too much from the theoretical viewpoint. I wanted to have the possibility of looking inside real problems and trying to get answers to big problems. And so we mentioned that I was part of the Human Microbiome project in 2010, more or less. And when I was part of it, the first output of the project was the sequencing data. So coming back from what we discussed now, this raw sequencing data that was generated, nobody really knew how to look inside them. Mm-hmm. There was huge amount of data, sequencing data, unstructured. No biologist could have looked into that. You need to have a very data-driven approach at that. So I was in a lab. My PI at the time, my supervisor, Curtis Attenauer, was also computer science by training, and we started to look at this data from the statistical viewpoint, the data structure viewpoint, and from there we start building stuff, and we saw that we were discussing, of course, with the microbiologists, and we found a common language there. To start discussing. So that was the key point. Having the computational background to be able to look inside the data and then discuss about what we are seeing in the data with experts in the field of microbiology, microbiome, and so on and so forth. And the thing I probably liked the most was that in doing that, I was also starting to learn microbiology and microbiome research. So now it's natural in my lab. There are microbiologists, there are computer scientists, statisticians, medical doctors, microbial ecologists, and it's natural. At that time, it was not natural, but that was, I think, what was the beauty of this of the path that mm-hmm. I did.
Interesting. And so before you mentioned that we were talking about fungi, but you mentioned that the microbiome that's present on food can be incredible. Probably, I would imagine there's striking differences between the microbes that are on one food versus another food. Presumably, they're eating that food or they're eating something in close proximity to it. I've always thought about our gut microbiome as, obviously it's influenced by what we eat, but I guess I've never really thought about the fact that maybe components of the microbiome actually come from the food itself, literally. Can you talk a little bit more about that? Like, what are some of the foods out there that have a big impact on our microbiome because it's it's actually shipping the microbes in rather than sort of feeding something that's already in there?
Yeah, if you think this is another way of thinking about transmission, this is a transmission in a One Health perspective. It's not person-to-person transmission, it's acquisition from the environment. So, and we eat the environment, if you like. So usually the other thing is important to say is that in many foods, microbes are not there just because they happen to be there, but they are a constitutive part of that food. Think about fermented food, for example. Right. Dairy products that are fermented food, like cheese. Cheese is a fermented food. Fermentation is different for different cheeses, but that is the key there. Or again, thinking about beers or yogurt or kefir and so on and so forth. These are all foods in which microbes are not just a side effect. These are the reason why the food is there, and also the quality and the properties of the foods are directly set in those cases by the microbes. So the same cheese done in different places, sometimes they have the same bacteria on them, but slightly different bacteria or slightly different regions in which the process is done means completely different bacteria. Some of these bacteria are completely unknown. Some are much well studied. Some are probiotic that are somehow characterized, and some are not. Some of these bacteria, they have the characteristics also to be able to colonize the gut, and others are not. So it is a very vast area of research, and we did a large study recently within a European project. It was, I think we sequenced something like 2,500 different foods, and we try to look at the overlap between these foods, microbiomes, and the gut microbiomes. This is a moving target. We are studying it, but I think the most fascinating aspect is again thinking about this in light of evolution. We co-evolved with our microbiome. We started to eat fermented foods thousands and thousands of years ago. Some of the microbes that are now only gut microbes could have been originated from food and then adapted to our gut through evolution.
Yes.
So even though it's like probably no more than 2-3% of our gut microbiome that is directly overlapping with our food microbiome, in reality, through the lens of evolution, this could be much, much higher than this.
I see. So yeah, when we think about this coevolution aspect to this, it's entirely possible that there were microbes in the past that were part of our environment. They lived on some food. They got into our gut to some extent, but then over time, they just became adapted and actually evolved and mutated such that they can no longer survive as well outside of our bodies and can only survive inside of our bodies. And then at some point, then if that becomes a vertical transmission from mother to infant, you could imagine situations where we used to have no relationship to a microbe. We start acquiring it from the diet, and then it effectively becomes an endogenous part of the body, even though it's not inside of us. It almost reminds me of the story of the endosymbiotic hypothesis of mitochondria. They used to be free living organisms, but then something happens such that it becomes totally embedded within our cells.
Yeah. And this is even more complex if you think about speciation of primates. Yeah. Because there could be a microbe host evolution in a specific primate, and then this primate is actually speciating. And then the microbe can be speciating themselves or not. But then in any case, even two primate species that are living in the same region, bacteria can jump from one to the other. So it's such an intricate network of evolution, transmission, and then seeding that I think is extremely fascinating to understand. To try to understand how the complexity of the microbiome right now is established by coevolution and all the other forces that are pushing on microbes. Again, in a One Health environment, because our microbiome is in the environment and vice versa, and actually we are in contact with the environment. Our skin microbiome is the first interface between us as biological entities and the environment we are living in.
Mm-hmm. What are some of the projects you're working on in the lab right now that are particularly exciting?
We are going much, much deeper into the axis I was telling you, so the diet, microbiome, health axis. And we are extremely excited to be able to reach now 200,000 individuals with very deep phenotyped diet, microbiome, and cardiometabolic health. This, I think, is really invaluable and is going to be exciting because it's more and more increasing these databases. And also, we have information and microbiome data for individuals that change their diet in time. So we can try to associate the change in diet with the change in microbiome and vice versa. And we also have clinical trials in this line of research, which are making things more and more relevant. We are also trying to isolate and cultivate some of the microbes that are completely unknown in our gut. And this is important because we may find new functions, also including function connected with positive diet and positive cardiometabolic health. So trying to close a little bit the thing there. Also, large part of what we do is connected still with computational methods to try to advance the field from inside. So when we study transmission, for example, transmission is possible. To study transmission is possible only if we are able to profile the strains at the single nucleotide level. Because it's only if we are able to identify that the strain in myself and in yourself are different, that when we find the same strain, then we can infer transmission. Then I was mentioning the field of oncology is extremely exciting there to think that we can have an impact for patients there. And there are a lot of microbial immunologists, mucosal immunologists with which is so exciting to interact.
Yeah, that's an exciting area too for a couple of reasons. One, I mean, when you think about something like colon cancer, it's not a big leap to think the microbiome would be involved, given that's where much of the gut microbiome is in the colon. And two, I just did an episode recently with a couple scientists who did lipidomics and some clinical work showing that there's a very clear link between dietary fatty acid composition that someone is eating and the amount of inflammation and colon cancer risk that they have in the gut. And those scientists were very excited about the role of the microbiome there. And it makes a lot of sense that there would be some connection because presumably the microbes are processing and creating new fatty acids to some extent based on the ones that we eat. So it's going to be really exciting to see what kind of diet microbiome interactions are there.
Yeah. So we are already at the point in which a microbiome test can tell with reasonably high accuracy whether someone has colorectal cancer or not, which is important because we can use it for screening, non-invasive screening, of course. And then, of course, this is one example in which a microbiome test is actionable because the action will be do a colonoscopy to investigate more, or you don't need to go to do a colonoscopy with high urgency and so on.
The microbiome provides a signature that you can use to decide if you need to do something more invasive.
Exactly like that. But this is screening for someone already having the cancer, which is probably colonized by microbes and so on and so forth. The next step will be to investigate the risk factors. And we actually have a big study. It's called Prospect, which is studying specifically early onset colorectal cancer, which is rising in the incidence. In young people, in young people here, meaning below 40 years old or 45, depending on the definition. And here the focus is on the risk factors, maybe the environment, pollutants, diet, and the microbiome. We are studying all of these to see whether the increase of early onset colorectal cancer is actually due to some of these characteristics, and then try to modify them to see whether we can lower it. And this is, I think there are several reasons to think that early onset colorectal cancer is specifically associated more with the microbiome than the other cancer, because otherwise it would not explain large part of this variability.
So we've covered a lot already, and this is a very rich and active area of research. Is there any final thoughts you want to leave people with, or anything you want to say to reiterate the topics that we talked about? Anything you want people to take away in terms of, practical things they can do to keep their gut microbiome healthy, or what they should keep in mind in terms of, what the big risks are out there?
Well, one thing I hope that came out from this discussion is that the human microbiome is extremely complex. Sometimes there is an oversimplification of this, but it's not the case. So we know still very little about the human microbiome. We don't know about the function. We don't know about a lot of function, at least. We don't know about how we can cope with this huge variability. The fact that the microbiome of each of us is unique has a lot of implications, because we cannot think about intervention, drugs, diet that can work identically to each of us, to all of us from the microbiome viewpoint, because the microbiome is unique. So I think the microbiome is at the same time a research field in which there is a lot of hype, but also a research field in which there is also a lot of unmet opportunities. And because for several reasons, the microbiome is a little bit, again, oversimplified, I would really like to that this information about the complexity and the fact that we are trying as a field to study this complexity before being able to deliver final results is what is actually going on, and should keep us busy for quite a bit of time still.
All right. Well, Nicola Segata, thank you very much for your time. Fascinating work, and I look forward to seeing what you come up with next.
Thanks to
Full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.